Hey, everybody. We got a chance to talk about a number of important ideas from the Chapter 6 PowerPoint, the notes from Chapter 6. Um, we talked about different types of transport across cell membranes, talked about diffusion and osmosis, transport through channel proteins, how some of those channel proteins can be gated so that the cell has some control over the movements of ions through those channels. And want to talk about a couple of other different types of transport now that we maybe didn't have as much time as I would have liked to in class. So, the difference between a channel protein and the carrier proteins that you see in the slide here um, are that the carrier proteins just have a little bit greater specificity and they tend to interact with whichever molecule is being carried across the membrane in a little bit more intimate way, often binding the molecule, going through a transformational change, change in structure, and having that conformational change result in the molecules being moved from one side to another. When a process like this takes place, like we see in the diagram here, when it takes place without the expenditure of any energy, then we would call that facilitated diffusion. Um, Molecules moving from an area of higher concentration to an area of lower concentration. Um, and there are lots of carrier proteins that do this type of facilitated diffusion. And there are also carrier proteins that do other types of transport, and we're gonna talk about that in a minute. But first, I want to look at a specific example, and we did touch on this in class, but I wanted to make sure everyone understood because this is, for me, a really big deal. Um, it explains how a cell can respond to the different environmental circumstances that it has to deal with on a regular basis. So, our cells have the ability to take in glucose. Glucose is a source of energy that pretty much every cell in the body would be happy to get a hold of, taking in glucose so that the cell can do cell respiration, use the energy from that sugar from the glucose to make some ATP to power the various processes that are going on inside the cell. But there are times when our body would not want cells to take glucose, uh, actively to take glucose up from the blood. There are times when we would want the cells to leave the glucose alone. If we don't have very much of it in our blood, then the body would prefer that the glucose is reserved for our brain, for our muscle cells, for the organs that might vitally need glucose on a moment-to-moment basis. And so this is showing how a cell could be differentially permeable to glucose depending on the signals it's getting from outside. So it shows at the top a cell that has no carrier proteins for glucose plugged into the cell membrane. And there may be circumstances where this is true, where there are absolutely none, but more than likely there may be a few of those glucose transporters in the membrane, and the cell might want to add some or take away some, depending on the circumstances. And so when this cell receives a signal that there is plenty of glucose available outside, available in the blood and the tissue fluid, then it would move some of the carrier proteins that are sort of lying in wait, embedded in a vesicle. And remember that these vesicles that we see inside of cells, they're made of the same lipid bilayer that the plasma membrane is made of. And it looks like the process of exocytosis when this vesicle moves up, migrates up to the plasma membrane, fuses with it, and by doing so, basically injects all of these glucose carriers into the plasma membrane. Now the cell's capacity to absorb glucose is greater than it had been before. The cell can take up more glucose from the surroundings. And if a cell wanted to decrease its capacity for taking up glucose, if it wanted to slow down the uptake of glucose, then it would just do the reverse of what we see in the top diagram. Instead of plugging the vesicle into the plasma membrane and thereby injecting all of those glucose transporters into the membrane, it would do what looks like endocytosis, and it would pull the membrane in and create a new vesicle inside the cytoplasm where all those glucose transporters can now be stored and sort of kept in reserve until such time that the cell needs them. And it's chemical signals like the hormones that are described in the text here, insulin and glucagon, that tend to cause these types of changes in a cell. Could be a hormonal signal like insulin or glucagon. It can also be signals that are being transmitted through nerve impulses. It could be signals transmitted by other types of molecules other than hormones. Molecules that are being secreted by neighbor cells, for instance. Those are called paracrine signals. And this is just showing endocytosis and exocytosis, a review of that. And again, the top one looks like the process that I was just describing. If you imagine that there were some transport proteins plugged into the membrane here, and this piece of membrane folds in, this invagination finally pinches off. If there were transport proteins in that membrane, now they are not in the plasma membrane anymore, but embedded in that vesicle wall inside the cytoplasm. All right, active transport is when we use energy to move molecules against their concentration gradients. So there may be lots of conditions where we want to get a higher concentration of a substance inside or outside the cell than the other side. And the diagram and text here is describing the active transport of calcium out of cells. And there are lots and lots of cells in the body that do this. The majority of cells pump calcium out. And calcium is a really important signal molecule in cells. And so we actively pump it out, and then we let it back in as a means of signaling inside the cell. And we'll talk about some different ways that it happens later on, but I'll let you pause this if you want to take a closer look. But this is an example of active transport where the movement of the substance across the membrane is against the concentration gradient from a low concentration to a higher concentration. And the process requires energy, and almost always that energy is ATP. And the last thing in this little video is to talk about one of the most important, arguably the most important active transport pump that exists in our body, and it's called the sodium-potassium pump. For those of you who have taken a biology class before, especially if you're in AP Bio, or for those of you who've done AP Psych, you have heard of sodium-potassium pumps, I'm pretty sure. And these are the pumps that take sodium ions from the cytoplasm and pump them out, and they pump potassium ions from the extracellular fluid into the cell. And the working of the sodium-potassium pumps is so important. It's been estimated that a quarter up to a third of the energy that our bodies used that our bodies use when we are at rest. So if you're just sitting around, about a third of your energy goes to running these pumps, and these pumps are responsible for keeping our nerve cells and our muscle cells functioning properly. If they're not working, then the nervous system and the muscular system are not working. Okay, so I said I would come back to the sodium-potassium pumps and say just a bit more about them. I'm not going to say more about them. You've listened to enough of me, and I typed it all on this slide anyway. So I'm going to let you pause the video here and read what is on the slide. But the sum total of the activities of the sodium-potassium pumps and the ion channels that you see on this particular membrane, and this could be a nerve cell, or in fact it could be a muscle cell as well, because virtually the identical things are happening on muscle cells. This is how we maintain a voltage, an electrical potential difference across our nerve cell and muscle cells through the operation of sodium-potassium pumps and some other ion channels. And this gives our nerve cells and our muscle cells the ability to respond quickly and generate an electrical impulse that can be quite useful if we want our muscles to contract, or if we want to send a signal across the body really, really fast. So pause this, take a look at this stuff. If you are a little baffled by all this information here, please write down a question. Please write down a question mark in your notes, something, and ask next time when we're in class.